when Wildtype started, there was no cell lines available for us to develop media for. So we really had to start from the very beginning and establish cell lines. So as we were establishing these cell lines, we were also trying to understand how to grow them, what is the right media formulations. And we learned that the pH for them, for example, is very different from mammalian cultures. My background was mammalian culture, so it was very surprising for me to see the temperature range is very different from them. And we had to figure out like what plating densities, what surface works for them. So it was a lot of learning.
And from the very early days, even establishing cell lines, we were taking advantage of the publications that's already there from academia in the 1960s. We were very lucky when we selected salmon because there were those papers existent. And we took full advantage of that, of temperature, of buffer systems, of even the starting media. There were papers for salmon in that space. And then more recently, we did RNA-seq analysis, and we compare RNA-seq results coming from culture that is with higher serum versus culture that's with lower serum, and compare the difference. Overall, we're looking at the difference in gene expression from these two different culture systems and trying to look for guides on which pathway should we target. We were only able to do this because of the help from the University of Victoria, even though we're not directly collaborating, but this university published the whole genome sequence result for coho salmon, as well as actually whole genome annotation for it. We would love to do the same of analysis for some other amazing seafood species like bluefin tuna, spot prawn, or other seafood. We have the cell lines established, but we actually today couldn't do the same RNA-seq analysis for those kind of species or cell lines because, as you can see on the table on the right, for bluefin tuna, at least there is sequencing result, but annotation is much less polished.
It all starts off with a seed. This is a sample taken from an animal. Goes into the machine here. The seed is then slowly cultured, which produces more and more. It's fed nutrients to keep growing en masse, at which point it's taken into the next machine. It's fed more nutrients. It gets bigger and bigger. If you can imagine feeding a sourdough when you're making bread, it's the same principle. The more you feed it, the bigger it gets. The next nutrients go into the next tank, and then the next tank. Eventually, when there's enough mass in the final tank, you end up with craft meat. From there, there's a little machine at the end, which is called a centrifuge. It's piped into there where the water is spun out, and we end up with the final product, which ends up in a piping bag in my restaurant.
I'm going to make a cocktail today called the Forged Highball. And this little number is a cultured Japanese quail tart. It's a perfect little accompaniment for the Forged Highball.
And with Wildtype, this is a really cool third restaurant that they're launching in at Robin in San Francisco. This is where I tasted Wildtype salmon when I talked about it a number of videos ago. So I would personally recommend this restaurant.
I'm Adam Tortosa, the chef and owner of Robin in San Francisco. Robin is a San Francisco-style omakase restaurant. I grew up loving to cook with my family. I also grew up loving to fish, so I think the combination of those two is what led to me becoming a sushi chef. I just remember as a kid being able to go out fishing with my dad and catch a bunch of fish, which is just not the scenario anymore. I want my son to be able to go fishing in 20 years from now. I think if we continue what we've been doing, that option is not going to be there for him. There's not enough fish. Our seafood industry today is just heading in a scary direction. The quality of fish, the quantity of fish is all just harder to come by. It's just simply not sustainable. The decline in our ocean is because of overfishing. Wild-type salmon is a new option beyond wild fish, not farmed, but something that is completely different. I don't think wild-type is going to replace farmed fish or wild-caught fish. It's one tool to help us get to a sustainable future where there are fish in the ocean. I'm super excited to put it on our menu. The thought of being like one of the first restaurants ever, period, is super exciting. The way I would describe wild-type taste is the same as regular salmon. If it didn't taste like salmon, we wouldn't consider it.
Okay, what's in wild-type salmon? First off, yes, it's real salmon made from real fish cells. So if you're allergic, steer clear. Salmon cells don't know how to be sashimi. They need structure. We use plants to give structure, specifically potato starch, soy protein, and cognac. It's a root vegetable. In the ocean, salmon eat fats made from algae. We use that too, along with other vegetable fats. Also, wild salmon color come from beta-carotene found in krill. Ours comes from plants, specifically a natural antioxidant called lycopene. For more information, please visit our website or reach out to us directly. Keep those questions coming.